binary fission in amoeba and budding in yeast and hydra with the help of prepared slide. So we will study about asexual reproduction in amoeba, yeast and hydra. First we will talk about binary fission in amoeba. Children, as we all know that amoeba is a unicellular organism, and just like a bacteria, it reproduces through binary fission. After replicating its genetic material through mitotic division, the cell divides into two equal-sized daughter cells. In this method, two similar individuals are produced from a single parent cell. Let us see what are the materials required. For this study, a prepared slide of an amoeba is required and a compound microscope is required. Let us see what is the procedure. Take a permanent slide of an amoeba and place it on the stage of compound microscope and adjust the mirror of the microscope carefully until the image achieves maximum clarity. Observe the permanent slide under the low power lens and then high power lens of the compound microscope. Move the slide very carefully back and forth to observe the different phases of binary fission in the amoeba through the lens of compound microscope. So this is the low power lens of compound microscope and this is the high power lens of compound microscope. So initially in low power lens, the structure, the permanent slide looks somewhat like this, and under high power lens of compound microscope, the slide looks somewhat like this. Let us see the observations. Initially, mother cell of the amoeba elongates and its nucleus divides into two through the process known as karyokinesis, that is the division of nucleus. After the nucleus divided into two, the process of cytokinesis takes place, where the mother cell cytoplasm splits into two daughter cells. This leads to the formation of two daughter amoeba cells. Each daughter cell have nucleus, develops into two organelles and grows into an adult organism. What is the conclusion of this study? This slide shows the nucleus of an amoeba. In the process of division, body of amoeba elongates and has a constriction in the middle. Hence, we can conclude that the slide shows the asexual reproduction process of binary fission in an amoeba. Let us start with the next slide study, that is budding in yeast. Budding is a type of asexual reproduction in which a new organism develops from an outgrowth or bud due to cell division at one particular site. The small bud-like projection coming out from the yeast cell is called a bud. What are the materials required? Prepared slide of an yeast and a compound microscope. Let us see what is the procedure. Take the permanent slide of budding in yeast and place it on the stage of compound microscope. Adjust the mirror of the microscope carefully until the image achieves maximum clarity. Observe the permanent slide of budding in yeast under the low power lens and then high power lens. Let us see what are the observations. We can observe that the yeast cells are spherical and oval in shape. First, a small protuberance appears on the body of a parent cell known as bud. We can observe these buds in the permanent slide of budding in yeast. We can also observe a chain of bud on the parent yeast cell. We can also see the nucleus of the parent yeast cell that is split into two daughter nuclei and migrate into each outgrowth. The bud detaches from the mother yeast cell by the forming of constriction at the place which can be observed. A yeast cell has a nucleus, vacuole, cytoplasm and the cell membrane. So this kind of structure we can observe under the compound microscope. The next study is budding in hydra. In hydra, a bud develops as an outgrowth due to repeated cell division at one specific site. These bud develops into tiny individuals and when fully mature detaches from the parent body and become a new independent individual. Let us see what are the materials required. A prepared permanent slide of an hydra and a compound microscope. What is the procedure? Take the permanent slide of hydra and place it on the stage of compound microscope. Adjust the mirror of the microscope and observe that permanent slide of hydra under the low power lens and then the high power lens of the compound microscope. Now move the slide very slowly back and forth to observe the different phases of binary fission in hydra. We can observe different phases of budding in hydra. Let us see what are the observations. Hydra produces a protuberance on the lateral side of the body wall. The protuberance later develops into young hydra by sharing body wall from parent hydra. The bud increases in size and volume by the division of cell. After its development is complete, the bud detaches from the parent body and starts an independent life. So this is the complete study of asexual reproduction in amoeba, in yeast and in hydra. So children, I hope whatever we have learned in our lab activity, you got each and every step clearly. So you have to write this identification study in Science Lab manual. And very soon we will meet again. Till then, have a nice day and thank you.